Now we're going to look at inheritance at the smallest level of the specific DNA molecule. So this is going to be DNA replication. The first thing we need to do is understand what DNA is. And when Thomas Hunt Morgan showed that the inherited characteristics were passed on by chromosomes, this gave us two options for what is the actual genetic material. So chromosomes are made up of DNA, which is our nucleic acid, and proteins called histone proteins that are used for wrapping the genetic material up. So one of these had to be the inherited information that got passed on from one generation to the next. But at the time of Morgan, we didn't know whether the genetic material was the DNA or if it was the protein. Most people at this time actually thought that the proteins, because there were so many more of them, or varied proteins, was what was the genetic information and not the DNA. So there are several researchers that went through the painstaking process of kind of proving to the scientific community that it was in fact the DNA that is our genetic material. So the first one, these are underlined because you need to know these scientists, was Frederick Griffith. He provided the very first evidence. This is 1928. You don't need to remember the years, just the names. He provided the first evidence that DNA can transform an organism, giving it kind of the first idea that DNA might actually be the information or the genetic material. So he did several experiments. He took a pathogenic bacteria. This is the S cell. And he put it into a mouse, and the mouse dies, which makes sense. That's a pathogenic bacteria. Then he took the same bacteria that was non-pathogenic, so R is non-pathogenic, and that means it's not disease-causing. And he injected that bacteria into a healthy mouse, and the mouse lives because it did not get a disease. Then he took the pathogenic bacteria, so those are the ones that cause disease, and he heat killed it, meaning he cooked it. This is kind of like the same thing you do with your food. If you had a dangerous bacteria in like salmonella in your chicken and you cook the chicken, it should kill the bacteria. So he put the dead bacteria into the healthy mouse, and the mouse lives. Happy mouse. Then what he did is he mixed those two. So he mixed the living non-pathogenic that did not kill the mouse and the dead pathogenic, which did not kill the mouse. And when he put those two together into a vial and put it into the mouse, all of a sudden the mouse dies. So what happened? What caused the mouse to die? We took two different experiments that did not kill the mouse, but when we combined them together for some reason, all of a sudden now they're pathogenic and they kill the mouse. So what happened is from the mouse, he cultured living pathogenic bacteria that were exactly like the original experiment that killed the mouse. And this showed that these changed. These living bacteria that he had put into the mouse had to take up foreign DNA from the dead bacteria, and it actually transformed them from non-pathogenic, meaning non-disease causing, into pathogenic, meaning disease causing. So this was the first idea that bacteria could assimilate or pick up foreign DNA from their environment, and that's called transformation. The next step came about 30 years later with Alfred Hershey and Martha Chase, and they were the first ones to really show that DNA is the genetic material, it is not the proteins. So they used something called a bacteriophage, which is a virus. So that's this guy here. It looks a little like an alien. Bacteriophages are made of two parts. They have a protein coat, which is this outer coat out here, which is made of protein. And they have DNA inside. So just like our chromosomes, they're made up of protein and DNA. So what they did is they let these bacteriophages infect some bacterial cells, and they just tracked which parts actually get into the bacteria to infect the cells. So when they labeled the protein, they noticed that the labeled part did not go into the cell. So the protein did not get inserted into the bacteria that were being infected. But in the second batch where they labeled the DNA, they noticed that the bacteria did get infected with the labeled DNA. So in this case, it went into the cell. So they were able to conclude from this that the injected DNA from the virus encoded the virus's genetic information, and that's how the virus was able to kind of take over these bacteria cells. And so this was the first kind of solid evidence here that the genetic information came from DNA rather than proteins. About the same time, there was another researcher named Erwin Chargaff who was analyzing the DNA of lots of different animals. So he came to a couple of conclusions, is that different species, so humans, chimps, dogs, rats, have different compositions of DNA. What he was doing was analyzing the amount of the different bases. So what he noticed is that the number of adenine was always equal to the number of thymine, and the number of guanine was always equal to the number of cytosine. Even though those numbers were different in the different species, A was always about the same as T, and G was always about the same as C. So this means if you know the percentage of a single nucleotide, so in this practice problem here, we know that adenine makes up 36% of the DNA. You can actually figure out the percentage of all of the other bases. So we know adenine is equal to thymine, and there are only 4 bases, guanine and cytosine. We know that all 4 of these make up all of the DNA, so they have to add up to 100%, right? That's all of it. Adenine plus thymine equals 72%, and we know it needs to add up to 100. So 100 minus 72 is going to give us 28% of the DNA that's left. So 28% of this DNA has to be made up of guanine and cytosine. But we know that guanine and cytosine have to be equal, so we can just divide this by 2 and get that both of these are going to need to be 14%. So in a question like this, if I give you adenine, you should be able to figure out the percentage of any of the different nucleic acids. So go ahead and try this next one. If cytosine makes up 27% of the DNA, what percentage is thymine? So now that we knew that DNA was the genetic material, and we knew a little bit about its bases, we knew that it had adenine, guanine, cytosine, thymine, we knew that it was made up of phosphorus and sugar, but we didn't know its physical structure, how it was actually put together. There were several researchers who were working on this. We have Watson and Crick, as well as Maurice Wilkins and Rosalind Franklin. And Wilkins and Franklin were using something called X-ray crystallography to study structure. So this is where you would crystallize the molecule and then you would shoot X-rays through it and then take a picture of the pattern that shows up after. So Rosalind Franklin is famous for producing this picture of a DNA molecule using this technique. And what happened is Watson and Crick were men, of course, and they were visiting Maurice Wilkins. And Wilkins actually gave this picture to Watson and Crick without her knowledge or consent about it. And so the reason that Watson and Crick were able to figure out the 3D structure of DNA is because they essentially stole this picture from Rosalind Franklin. And then Franklin went on to die before they were given the Nobel Prize. And so Wilkins, Watson, and Crick all received a Nobel Prize for their discovery of the structure of DNA, but Rosalind Franklin doesn't usually get the recognition that she deserves for actually discovering this helical shape. So before Wilkins gave Watson and Crick this photo, Franklin had already concluded that the outer sugar phosphate backbones were on the outside in a double helix structure as indicated from this molecule's picture. And she thought that the nitrogenous bases were paired on the inside of the structure. Watson and Crick actually had this backwards. They thought that the phosphates and the sugars were on the inside, and then the nitrogenous bases were kind of open and on the outside. Watson and Crick also thought it was a triple helix instead of a double helix, and so they were able to kind of put all of this together once they had taken this photo from Rosalind Franklin. So Watson and Crick built a model of the anti-parallel structure of DNA. So here you can see the structure of DNA. Here's one nucleotide. All of the DNA nucleotides are made up of a deoxyribose sugar, a phosphate group, and a nucleic acid. And there are 4 different nucleic acids. Adenine and guanine are purines. So adenine and guanine here are called purines. They have 2 rings, so those are purines. Thymine and cytosine are pyrimidines, so thymine here and cytosine. So what you'll notice is that a purine always pairs with a pyrimidine. We know that A always pairs to T with 2 hydrogen bonds. Those are my hydrogen bonds here. And G always pairs to C with 3 hydrogen bonds. The way to remember this is AT2. They both have a ta sound. And GC3, they rhyme. Notice that the left side of the DNA is upside down from the right side of the DNA. So they run something that is called anti-parallel. There is a 5' end, and the 5' and the 3' comes from numbering carbons. When you get to organic chemistry, you'll learn how to number carbons. So on a carbon molecule here, this is the first carbon, second carbon, third carbon, fourth carbon, fifth carbon. So the 5' end is named because the phosphate group is attached to the number 5 carbon. So that is the 5' end of the DNA. Notice on the left that the 5' end is over here at the top, and on the right, the 5' end is over here on the bottom. They run in opposite directions. The 3' end has a hydroxyl group on the number 3 carbon. So again, 1, 2, 3, 4, 5. So this is the number 3 carbon, and it has a hydroxyl group on the end. So this is called the 3' end. So on the left, the 3' end is on the bottom, and on the right, the 3' end is over here on the top. So on this side, it runs 5' to 3' down, whereas on the right side it runs 5' to 3' up. So they run 5' to 3' in opposite directions. So that means anti-parallel, and this is going to have a lot of implications on how the DNA are actually going to replicate. So at first, again, Watson and Crick didn't have the pairings right. Even once they figured out what the pairings were supposed to be, they thought that A paired with A and G paired with G and so on, but that didn't result in a uniform width. So if A paired with A, remember A is a purine, then it would be really wide. And the width of the DNA helix would be all the way out here. There's just too much stuff in between those. Pairing cytosine with cytosine created something that was too narrow. So you'd end up with like the structure of DNA looking like a wonky hourglass here. What they eventually realized is that a purine always has to pair with a pyrimidine in order to be consistent with the X-ray crystallography structure, which said that there was a uniform width to the DNA. So using Chargaff's rules, where we knew that A was always equal to T, and G was always equal to C, it made sense that the purines always paired with the pyrimidines, and that it was always specifically in this order. So again, here you can see the pairing of adenine to thymine using 2 hydrogen bonds, and you can see the pairing of guanine to cytosine using 3 hydrogen bonds, and that this correlated with Chargaff's rules. Watson and Crick, in their second paper, noted that this specific pairing made it really, really convenient as a copying mechanism for the genetic material. So DNA could copy itself with only half a molecule. So if we only had one molecule of DNA, we could figure out what the other side should be. So the genetic material is going to be able to always pair. We know that if we see an A on one side, there has to be a T on the other. If we see a T on one side, there has to be an A on the other. G is always paired with C, C always paired with G. And so if we only had a single strand of DNA, it was totally possible to come up with the other strand of DNA and have the correct DNA. And so this suggested a mechanism for how DNA copied itself in DNA replication. So the two strands are called complementary, meaning that they have complementary strands. One acts as a template in order to build a new strand. So we take our DNA molecule that is twisted up. We need to unwind it into something that can be opened, and then take those two strands apart. And then once the two strands are separated, a new strand can be built using each of the original strands as templates until we have two exact copies of the original DNA. So we've got a parent molecule. Again, look at the anti-parallel nature, 5' to 3' in opposite directions. And you can see that they are complementary. A pairs with T, C pairs with G, A pairs with T, A pairs with T, C pairs with G. This is called the parent molecule. So this is the original DNA molecule. At some point during DNA replication, it had to separate. These are hydrogen bonds here, right, holding these together. So these are actually pretty easy to break. They're very weak associations. So we can break this section of DNA into two parent strands or template strands of DNA. And then, one at a time, new DNA molecules can be added. So an A knows that we need to put a T here, and a G, and an A, and a T, and a C. And so what we end up with is two brand new strands where each of them is 1/2 from the template, or the original parent molecule, and the other half is new. And if you look at these two new strands that we made, they are exactly identical to the original strand, and it's just based on complementary base pairing, A to T, G to C. So this was Watson and Crick's model of replication, and it was called semi-conservative model. And it predicts that DNA replicates exactly the way that we just went through, that each strand will conserve one strand from the parent and then have one newly made strand. At the time, there were several other theories or models of how DNA was being replicated. There was the conservative model saying that the two parent strands actually copied the new strands, and then the original parent strand rejoined back into the original parent, and so that the conservative model would result in strands that were completely old and completely new with no mixing of them. And then the dispersive model thought that each strand was a complete mix of old and new DNA. And so Watson's model was based on logic, but they needed to show that this was true. So Michelson and Stahl performed an experiment on cultured DNA to show how the DNA was replicating. So what they did is in the parent strand, they used a heavy isotope. So this heavy DNA is going to weigh more for the parents. And then they replicated it twice using a lighter isotope. So the new DNA would weigh less. And then they centrifuged it after each replication to see where the weight of these DNA bands were. So the heavier it is, the further it goes to the bottom. The lighter it is, the further it's going to go towards the top. So here's an example of what we would expect from the semi-conservative model. So here is the parent or the old generation. Here's the first replication and then the second replication. So the parent would have been very heavy, And then in the first replication, each of these strands has one strand from the parent, which is heavy, and one strand from the new DNA, which is light. So in a centrifuge tube, both of these strands look the same. They're going to weigh the same. So instead of having a heavy strand, we would get one line here about in the middle. After the second replication, each of these would split apart and make two new ones, whereas this one has one strand from the original parent, and this one's actually going to have two new strands. So a centrifuge for this one would have a line at the same height as the first one for these two, but then these two strands are lighter, so it would get a lighter line up here towards the top of the centrifuge tube. What they expected to see if the conservative model was correct was that, again, we start with the same heavy parent strand. The conservative model says that it copies it and then each of the parent strands go back together. So on the first replication, we would actually see a really heavy and a really light strand. So this one would be heavy at the bottom, this one would be light at the top. And then the second replication, we would see the heavy strand again at the bottom and three lighter strands up here at the top. So this one would probably just be a little darker. So this is what they would have expected to see if the conservative model had been correct. And then the dispersive model was the idea that kind of the old parent DNA got broken and evenly dispersed amongst all of the new DNA. So in the first replication, each of these would be about half old and half new. So they would probably be one line here in the middle. And then each of these in the second replication would be about 3/4 new and 1/4 old. So these are all going to weigh the same as each other, but they'll be a little bit lighter. And what Meselson and Stahl saw is that this was actually the pattern of centrifuge results, where they had one line on the first replication and two lines after the second. And so they were able to eliminate the conservative as well as the dispersive model and say that Watson and Crick's semi-conservative model was in fact the way that DNA was replicating.